So good afternoon to everyone. Today we are going to discuss about that new topic, the vaccine drug delivery system. The vaccine means everyone knows it is a way we can administer the drug through oral, topical, in that parenteral. So in that nasal route we can administer the vaccine delivery of drugs. Let's move to that one by one. There are the several ways we can administer the drug into the body to improve our immunity. There are the several subheadings in the topic like introduction, types of vaccines, delivery system for vaccines, uptake of antigens, single shot vaccines, mucosal vaccine drug delivery systems, and the transdermal based vaccine drug delivery systems. So in that, what do you mean by vaccines? Vaccines na enna? Vaccine is a material that induces a immunologically mediated resistance to the distance to the disease, but not necessarily an infection. The vaccine are generally composed of killed or attenuated organisms or subunits of organisms or DNA encoding the antigenic protein of pathogens. The term of vaccines applies to biological preparations produced by living microorganisms that enhance the immunity against the different types of disease, either prevent or treat the disease. The traditional vaccines administration faces the many challenges like need of cold chain storage, that means to store with the cold storage only, then multiple doses for efficacy and patients hesitancy due to the needles, okay, because of that the parental administration. The modern vaccine delivery systems is being developed to address this issue. So there are some common and traditional vaccines that has to be developed and enhanced the new technology. So their primary goals are to protect the antigens, they shield the vaccines from the degradation in the body, enhance the uptake, deliver the antigen specifically to the immune cells for the stronger response, a controlled release, provide the sustained or timed release to mimic the booster shoots, improve the stability to reduce or eliminate the need for cold chain storage, so we can improve the stability of the dosage home, enable the needle free delivery, improve the patient's compliance and safety. Let's move to the next slide. The vaccination is the most efficacious and valuable tool in the prevention of infectious disease. The provided that there are administered prophylactically in anticipations of pathogen exposure. So the goal of the vaccination is to stimulate the strong protective and long-lasting immune response to the administered antigen. So there are the several properties of ideal vaccines. First one is it gives the long life immunity, prevent the disease transmission, like COVID vaccines which we are administered, okay, like PCG. So likewise several examples of vaccines are available. So rapidly induce the immunity, stable and chief and shape, effective in all subject, means it is very effective for the old peoples and the younger peoples that transmit the maternal production to the fetus. There are the various routes of administration. So we can administer to the various routes like the deep subcutaneous or intramuscular route. So most of the vaccine which we are administered to the intramuscular route only. Then oral route, that oral BCG. Then transdermal route, the transnasal route, live attenuated influenza vaccines. Then sacrifications, that is a smallpox vaccines. There are the composition of vaccines. The vaccines consists of bulk antigen plus other fluids like water or saline, and included the preservatives and adjuvants are present. That is a basic components present in the all the vaccines. These ensure the quality and the potency of the vaccines over its shelf life. The vaccines are always formulated as to be safe and immunogenic when injected to the humans. It is formulated as a liquids but may be freezed for like reconstitution immediately the period to the time of injections. It should be available in the form of freeze-dried product for the reconstitution because of the stability problem. Then there are several steps involved in the vaccine action. So when inactivated or weakened disease causing microorganisms enter into the body, they initiate the immune response. This immune response mimic the body natural response to the infection. So this is the basic steps involved in the working of vaccines. How the vaccine is working in our body, the pathogen attenuated, then the antigenic determinant, which we are added adjuvants, then finally inoculation. When it is entered into the memory cells of our body, then produce a long-term immunity. Then the classification of vaccines. The vaccines may be classified in the several way like traditional vaccines and innovative vaccines. The traditional vaccines means which we are used in the long days ago, like killed vaccines, live attenuated vaccines and toxoid vaccines, some unit vaccines. Then innovative vaccines, these are the recent development, that is a conjugate vaccines, recombinant vector vaccines, T cell receptor vectored vaccines, the valence and heterotypic. So traditional vaccines these are killed but previously virulent microorganisms that they have been destroyed with the chemicals, heat or radioactivity or antibiotics. For example, influenza, cholera, polio vaccines, that is a killed vaccines. Live attenuated vaccines, some vaccines containing live attenuated microorganisms. Some of these active viruses that have been cultivated under the condition that disable their virulent properties that produce a broad immune response. For example, the yellow fever vaccines and measles vaccines. Then we'll see the toxoid vaccines. It made from the inactivated toxic compound that causes the illness rather than the microorganisms. The toxins are inactivated by treating them with the formalin. Okay, for example, chemicals such as detoxified toxins are called toxoids. For example, tetanus and diphtheria. Then subunit vaccines. It is a protein subunit. Fragment of it can create an immune response. So instead of that entire microbe, the subunit vaccines include only the antigens that stimulate the immune system. So for example, the plague immunization. Then let's move to that innovative vaccine. This is some recent vaccines which we are currently available. The conjugate vaccines, the certain bacteria have polysaccharides, the outer coats that are poorly immunogenic. By linking with the other outer coats to the protein, the immune systems can lead to recognize the antigen. For example, Haemophilus influenza type B vaccine, that is a conjugate vaccine. It is linking with the outer coats to the protein. Then recombinant vector vaccine, by combining the physiology of the microorganism and the DNA of other, the immunity can be created against the disease. For example, DPT vaccine. Then the valence, the monovalent and multivalent vaccines used to immunize against a single antigen. Multivalent means it is used to immunize against the two or more microorganisms. Only that is the difference between the monovalent and the multivalent. Okay, then heterotypic vaccines, the vaccines that are the pathogens of other animals that don't cause the disease. Okay, the vaccine that are the pathogens of other animals that don't cause the disease. For example, the BCG vaccines made from the Mycobacterium bovis, the product against the tuberculosis. Okay, so the vaccines, the antigens, it is obtained from the Mycobacterium of living cells. The BCG vaccines made from the Mycobacterium bovis to the product against the tuberculosis disease. Okay, so the remaining continuation we will see the next class. So thank you for patient listening. Thank you.